Welcome to the ISV webinar series. And Dr. Menno Cox is going to be speaking and providing us an update on the development of the influenza vaccine. Menno is a well-recognized name for her contributions in the influenza vaccine. She is an outstanding scientist and a great manager. Mano had the distinction of developing and getting it approved, the first recombinant influenza vaccine. I think her bio was on the website. You have seen it. She is highly accomplished. She has a great combination of scientific leadership and also the management experience. So she has PhD and also the MBA. So that makes her much more different compared to the people like me who have only the PhDs. And another fact that I think I would like to share with you guys about Minnow in that she is an outstanding coach, mentor, and provides the guidance to the people, both the new people who are entering into the field and then try to make a career, and also the experienced person, people like me who are have a lot of experience, but I think I benefited from Menno's guidance and experience. So I think she is outstanding in multiple aspects. So without I can keep on speaking and I can take the whole hour to just talk about Menno, but I think that's not the point. I think I just wanted to make sure that you understand it, that she is highly accomplished and now she is the president for ISV organization. She was also the fellow for ISV. So at this time and point, I will invite open the floor for Menno to share her vaccine development, influenza vaccine development experience and the learnings with you. But before I hand over the microphone to Menno, I would like to let you know that I think probably you can post your questions and comments on the chat, and also you can raise your hand. And I think we are looking forward for a very exciting and interactive session. So without further ado, Menno, it's all yours.
Thank you very much, Indresh. I just want to say we obviously were expecting a talk by Indresh, and due to unforeseen circumstances, that could not happen today. So I proposed to Indresh to step in and actually do a talk together with him, because part of the work that I'm going to talk about today has really been led by Indresh as well. We both used to work together at Protein Sciences, and he did an excellent job in guiding the development of Flublok. So I'm going to try to share my screen right now and start this presentation. So what we want to do is today we want to talk about the development of a recombinant influenza vaccine. And I apologize to those of you who have already heard part of this story. And I will end up with some thoughts on what the implications of our trajectory were for and are for the development of a universal influenza vaccine. So at Protein Sciences, I basically spent about 20 years working on getting from an idea into a product. I must say that at the moment that I joined Protein Sciences in 1998, the company had already conducted a number of clinical trials with a monovalent hemagglutinin-based influenza vaccine, but we really needed to find a way to get from that, idea into an actual product that ends up in a vial. And what you can see in this in this presentation is basically, or in this slide, is basically a little bit of a promotion how we try to promote Flublok influenza vaccine after it was licensed by the FDA. We really put an emphasis on the fact that it had 3 times more active ingredient in the vaccine, even though we were not allowed to call it a high-dose vaccine. It was not produced in eggs. It didn't contain influenza virus, no antibiotics, and no thimerosal or other preservatives. But what I will do today is really go into the journey, how that got us there. And one might say that during that period of development, which took actually more than 20 years, we faced quite a few challenges. And those challenges were in product development, they were in GMP manufacturing, they were in clinical development, in getting that regulatory approval from the FDA, and then subsequently in distributions. And I probably do not have to convince most of you that developing a vaccine takes a lot of time and costs a lot of money. So if you look at the Flublok development timeline, we basically started with the first clinical studies in 1993. 9 studies were performed in collaboration with 9 human clinical studies were performed in collaboration with the National Institute of Infectious Diseases. And it was basically in 2008 that we were able to have sufficient material to conduct a biologics license or to submit a biologics license application. So it then took until January 2013 to actually get FDA approval. And the time, and I'm going to go into a little bit on what the challenges were. We had inspections, we received complete response letters, we had issues based on, toxicology, CMC, etc. So the CMC challenges were really big because an influenza vaccine is a vaccine that changes from year to year, and usually in a CMC package, chemistry, manufacturing and control package that you submit to the FDA, you include a lot of validation data. And if your product changes from year to year, it's not so easy to get that validation data together. But altogether, the whole time from doing our first human clinical study until FDA licensure took close to 20 years. And often when we hear, scientists speaking about presenting some early clinical efficacy data, they may not necessarily realize that it might still take a very long time before that will actually result into a product that is being delivered, that you can get in the pharmacy or in a doctor's office and being delivered to people. So very briefly, the recombinant influenza vaccine was based on hemagglutinin. Hemagglutinin is one of the surface antigens of the influenza virus. And as you can see in this EM graph, on the outside of the virus, you have spikes, and 80% of those spikes are formed by the hemagglutinin. The other 20%, nearly 20%, is neuraminidase, but we decided to focus on hemagglutinin because it was shown in already in various papers and animal models that antibodies against the hemagglutinin protect against influenza. And since the influenza virus is an RNA virus, it is highly prone to all kind of changes, and as a result of that, changes in that hemagglutinin require an annual update of the vaccine. The RNA virus has a segmented genome, and as you can see in this schematogram, we have the hemagglutinin on the outside, the neuraminidase on the outside, as displayed in these green areas, but there's also an M protein on the outside, barely visible, and then that's a small amount. So the baculovirus production system would be uniquely suitable for the production of this hemagglutinin, because then you wouldn't have to change the cell line every year. The only thing you would need to change was the insertion of the hemagglutinin in that baculovirus. So what we do, again, in a nutshell, is we engineer the baculovirus with the gene of interest, hemagglutinin. We insert that in the baculovirus. These baculoviruses are very specific to insect cells, and there is a very powerful promoter, the polyhedrin promoter, that generates high yields of protein, of your protein of interest. So the cell line that I just mentioned is derived from the fall army moth, and it is basically a cell line that was highly characterization of that cell line took about 2 years, but it's the same for any hemagglutinin that you produce. So these insect cells are being cultured in a bioreactor, in a fermenter. When they are in their logarithmic growth phase, you infect the cells with an engineered virus. This is then incubated for about 48 to 72 hours, dependent on the hemagglutinin that you want to produce. And subsequently, the protein, the hemagglutinin, is isolated. The protein forms rosettes, as can be viewed again under an EM microscope. And this protein is then purified to greater than 90% purity into a final product and formulated with PBS into a vaccine. We felt that getting Flublok approval would validate this technology. And again, this shows in a graph, like how do you do this? You can actually clone and express that gene and go from basically a DNA sequence, or sorry, I should say an RNA sequence, into a final product in 22 days. So very quickly, you use reverse transcriptase to make a copy of the RNA. You then insert that into the baculovirus, which is a double-stranded DNA virus. And that double-stranded DNA also ensures that you actually make in these insect cells the product as it appears in nature, versus trying to grow an influenza virus in a embryonated chicken egg, where you often get mutations so that the virus grows better in these chicken embryo cells. So after fermentation, we would use a whole series of purification steps to remove contaminants, including DNA and other proteins, other undesired proteins. But key, you could go from gene to production in 22 days. Now, clinical testing, and I took this slide from a New York Times article, and most of you will be familiar with this, is basically can be divided into 4 different stages. There's preclinical testing, that is usually testing your vaccine on cells and then give it to animals such as mice, monkeys, rabbits, to see if it produces an immune response. Subsequently, you start with human clinical studies in phase 1, which is usually a safety study. You give the vaccine to a small number of people to test the safety and the dosage. And of course, you will also generate some experience and some information whether the vaccine indeed induces or stimulates the immune system. Then you proceed to phase 2, expanded clinical studies. And here you generally give the vaccine to hundreds of people split into different groups. You might look in different age ranges, and you see whether the vaccine acts differently in those groups. And those trials obviously further confirm whether the vaccine is safe and whether it actually does what you expect it to do. And then in phase 3 efficacy trials, you usually include thousands of people, and you wait for how many people will actually become infected with the virus. You compare that between volunteers who receive a placebo, and these trials can then determine if the vaccine protects against, here it says coronavirus, because this was indeed to explain the coronavirus vaccine, but we obviously did that for influenza. And then you measure the efficacy rate. So the phase 3 trials are usually also large enough to reveal evidence of of rare side effects. So if you look at all the clinical trials that we did for Flublok, we basically most of the times compared Flublok to Fluzone, which was considered the golden standard vaccine. This is a vaccine made in eggs and has 15 microgram of hemagglutinin, whereas I mentioned before, Flublok contains 45 microgram of each hemagglutinin present. And what you can see in this slide in a simple way, these were trivalent influenza hemagglutinin studies when it says FB greater than FC, it basically meant that we were able to get a superior immune response, geometric mean titer, and seroconversion in 7 of the 12 instances where we tested. In one instance, Fluzone outperformed Flublok, and that was in a study in an elderly population for one of the B antigens. And the other situations, we had a comparable immune response. We also performed an efficacy study. We were able to do this study because in the early days, I think this study was conducted in 2004, the influenza vaccine was not yet generally recommended in the United States, and that enables you to do indeed a placebo-controlled study. And what you can see in this study, we had about 4,500 people, half of whom who received Flublok and half of whom who received the placebo product. And what we then did was we looked for positive cultures due to a strain represented in a vaccine, and we looked for positive cultures due to any strain regardless of match of the vaccine. And what I have to say is these efficacy studies are generally performed throughout an entire influenza vaccine season, I should say. So in other words, the influenza seasons could start as early as September and could go as long as March. So therefore, what you would do is we started vaccinating in September, and we then monitored performance of the vaccine all the way through April. And what was very obvious in this study was that in the first 3 months of the study, obviously you were way better off and you had like 100% efficacy of the vaccine. But as the season goes on and the immunity in people that were vaccinated wanes, you start seeing some breakthrough of infections. So we basically... and I'll start with the bottom here, because here we really had like a larger number of cases, because this was a season 2004 where there were a lot of viruses that were circulating that were actually not matched to what was included in the vaccine. So this bottom part of the graph is actually a somewhat more important part of the study. But what you can see here is that we had quite a few cases of influenza in Flublok, but obviously more in the placebo group. And we had an overall protective efficacy of 44.6% against viruses, against any influenza virus, whether they were included in the vaccine, yes or no. For type A influenza, that means H1 and H3, that protective efficacy was higher, was 54%, and for B it was only 23%, with the confidence interval crossing 0, which basically means that you cannot really make any conclusions on efficacy there. Now, if you look at matched strains included in the vaccine, you see that efficacy is much higher. And as I mentioned before, it's important to realize that especially early in the season, you would see actually a much higher efficacy than this. So Flublok was efficacious despite a suboptimal match in the vaccine. So I mentioned there were also some regulatory challenges, and those regulatory challenges, they basically related around the fact that there had not yet been many products that were made in insect cells that were approved. But in 2007 and 2009 in the United States, so 2007 in Europe and 2009 in the United States, Cervarix, the human papillomavirus vaccine from GSK, was approved by the FDA, and that was very helpful to us because it meant that the FDA had become a bit more familiar with the technology, with the baculovirus production technology, and that enabled them to actually go quicker, come forward quicker with the questions that they might have. And then in 2010, a second insect cell product was approved, and that was Provenge, which was a prostate cancer immune therapy product. And also later, even later, we saw approval of Glybera, which was a lipoprotein lipase deficiency product made using adeno-associated virus, basically gene therapy product approved by EMA. And then again, Flublok finally being approved on January 16 in 2013 basically made Flublok the first and still today the only recombinant influenza vaccine in the United States. And with these approvals, we were able to remove a barrier for insect cell-based production platform from a regulatory viewpoint. And maybe for the veterinarians under us, there's also, and today there are actually many more products approved, but there's also a PCV porcine circovirus vaccine approved by the USDA in 2006, which was produced at a 3,000 liter scale. So I guess the breakthrough in funding for protein sciences came when people started realizing that recombinant technology would be the pandemic solution, because recombinant pandemic influenza vaccines would have been available, could be available much earlier in a pandemic and not after a pandemic, as with the egg-based technology. And this was this slide is adopted from a presentation by Robin Robinson, who gave this at VRPAC in 2012. And here he shows basically the projected availability of a recombinant vaccine versus an egg-based vaccine. And Protein Sciences demonstrated that the first batch of the 2009 H1N1 pandemic vaccine, we harvested 4 days after declaration of the pandemic, and we released it 30 days later for clinical studies. So you can be really fast with a recombinant technology if you indeed know, how to purify that product. And I think that that was a challenge during the COVID outbreak. So as I said, this whole recognition that recombinant technology would play an important role in a pandemic was a breakthrough for protein sciences, and we've also later seen for the COVID vaccines, because the development of vaccines cost a lot of money. And when the PCAST report, a report issued by the President's Council of Advisors on Science and Technology, this report was issued in 2010, it basically also allowed BARDA to provide protein sciences with a very large funding that helped us to do further clinical studies that really convinced everyone that Flublok was not just an influenza vaccine, but was even an influenza vaccine with a far better performance than the other licensed vaccines. So again, the greatest potential for substantially shortening the timeline and increasing the reliability of influenza vaccine production lies in the use of recombinant DNA technologies. So BARDA provided $150 million in funding to support licensure of Flublok through a contract. And we also had an out-licensing strategy with partnerships for Flubloks outside of the United States, which provided also more than $100 million in funding and resulted in the licensure of Flublok by regulatory authorities in Mexico and Brazil. So in Japan, we had a collaboration with a company called UMN, in Mexico with Leomont, in Brazil with Origin, in Argentina with Synergium, and in the Middle East with Avanscape. And from all these collaborations, we didn't just receive money, but we also learned a lot of things that were necessary to, to get this product across the finish line. Because one of the things that I didn't really mention and emphasize enough, Protein Sciences had a very small but highly motivated team that basically worked day and night to get to this point. And at the moment we filed our BLA in 2008, we were actually a company of only 40 people large, and that grew over time to a little more than 100. So January 16, the good news, we received a letter from the FDA that's we were issuing a license to Protein Sciences for the production of Flublok. Now, there were a lot of challenges associated with this FDA approval that we received. The FDA only gave us 4 months stability, even though we had wonderful stability, we thought, but statistically it was very hard to convince the FDA to give us more than the 4 months. The presentation, as you could have seen in the first slide, was originally in vials, and a lot of vaccines on the market by the influenza vaccines on the market by that time had already moved towards prefilled syringes. Our product was trivalent, and the market was moving to a quadrivalent vaccine. Our indication was 18 to 49 years of age, and we all know that it's mostly the little kids and the older people that are most vulnerable to severe influenza. Our process had low yields. Here's, by the way, where Indresh came in and really became very helpful in improving those yields. Our scale was 350,000 doses is what we could produce. And if you imagine that the influenza vaccine market at that time was around 120 to 130 million doses in the United States alone, making 350,000 doses is not very much. And we had, of course, a distribution and sales challenge. So actually, the real work started after getting this license, because we now needed to really scale up our production so that we could make more doses. And in order to do that, we licensed a facility that was previously used to produce Prevnar in Pearl River, a Pfizer facility, and this facility was adjusted, and it was basically adjusted within 100 days to go from a bacterial production facility to actually make Flublok. And we also expanded the age range. So in other words, we provided enough data to the FDA that they said, okay, this product is now approved for people 50 years and older, and that was in Q4 of 2014. We then filed multiple BLA supplements to get the facility in Pearl River licensed, and we obtained that in 2015. And then in 2015 and 16, we proceeded with doing more clinical studies, post-marketing studies. We, for example, had one rare side effect in our clinical studies, and that made the FDA demand that we would conduct a 50,000 subject study to ensure that this condition was not associated with Flublok, but was actually an anomaly in the study population. So we also were able during this time to do a very large comparative study with an active comparator, and that generated some really exciting data that I will go into. So the expanded stability, as you can see in this slide, this slide just summarizes the multiple years and many years of work to address the challenges that we had. And again, even though stability already looked really good, as you can see in this left part of the slide, it was not good enough initially to give us the entire influenza season, but we generated a lot of data to actually ensure and to make sure that the FDA would give us an extended expiration date of our product. Also, the clinical studies here, I will show them in somewhat more detail later, so I won't go into that. We then made a switch in a formulation. We made sure that we also were able to supply Flublok in a prefilled syringe versus a vial. That required a lot of comparability and biocomparability. And we worked very hard on improving the yield and scaling up the process. Again, as I mentioned before, that is where Indresh and his team played a critical role. So the top line results of a quadrivalent Flublok study versus an inactivated influenza vaccine are presented here. Again, study conducted over the entire influenza season. So now we are not looking at placebo, but we're looking actually at an inactivated influenza vaccine. This is a study in over 8,500 people. We had, over the duration of the study, 58 cases of culture-confirmed influenza in the Flublok group versus 101 in the inactivated influenza vaccine group, which translates into an attack rate of 0.13% for Flublok and 0.23 for the inactivated influenza vaccine, meaning that the relative vaccine efficacy against culture-confirmed influenza in comparison with an influenza vaccine is 43%, and the relative vaccine efficacy against culture-confirmed influenza plus fever, which is a little bit, more aggressive criteria, was 40%. So when I say aggressive criteria, if you have CDC ILI, it basically means that you also have fever in addition to other respiratory symptoms. So we also looked at severe serious advents, and there were 19 cases of medically attended ILI during the 6 months after vaccination, 6 of which occurred in the RIV group and 13 in the inactivated influenza vaccine group. And hospitalizations for influenza, 3 occurred in the group that received the egg-based influenza vaccine and 1 in the Flublok vaccine group. So you can then split out this data further in all kind of other criteria, looking at different age groups and what the efficacy is there. As you can see, the efficacy is a little less. The comparative efficacy compared to a licensed influenza vaccine that is made in eggs is a little lower in the older age group. It also makes it different if somebody had received the influenza vaccine the previous season. This is work that was previously extensively described by Wendy Keidel from Baylor Institute, that if you receive an influenza vaccine multiple seasons in a row, that in principle you may experience waning immunity. It's clearly demonstrated in this study as well. And yet, the reason why it is still recommended to take the vaccine every year is that if you don't take the vaccine, your immunity is waned so far the next season that you might really not be protected at all. And again, we observed the same thing: good protective efficacy against, or good comparative efficacy against influenza A, also good against B, but not as good as influenza A. And again, RT confirmed, PCR confirmed influenza is a bit more sensitive than culture confirmed, but culture confirmed is basically, again, the golden standard. And that's why that is what you generally use to describe your clinical studies with. So the 2017 season, we were able to release 9 million doses between the United States and Mexico, because we have been able to get approval for the Fall River facility in 2015. We had been able to source material from Flublok from Japan in 2016. And, we were able to get a longer shelf life. As I mentioned before, we went up from 4 months to 9 months. And we were able to get a quadrivalent influenza vaccine approved. So a lot of progress with the protein sciences team in a relatively small time. But the distribution remained a major challenge, because even though we had data that Flublok outperformed the licensed influenza vaccines, that didn't translate in a larger uptake in the market. And Sanofi then completed the acquisition of Protein Sciences in 2018, because they had a sales force and they would be able to take our influenza vaccine to the next level. And I think that is, exemplified by the fact that they today are selling close to 10 million doses. During the Sanofi era, a post-marketing study was conducted in collaboration with FDA and Medicare during the 2019 influenza season. So this is a study published by Izurietti from the FDA, and the title of the article is Comparative Effectiveness of Influenza Vaccines amongst the US Medicare beneficiaries ages 65 and older during the 2019-20 season. And what they basically described, the main conclusion is that the recombinant influenza vaccine was significantly more effective in preventing influenza hospital encounters. And you see how large the study group almost needs to be to kind of demonstrate data like this. Recombinant influenza vaccine was also significantly more effective than all other vaccine types, including the adjuvanted inactivated cell-based influenza vaccine in the inpatient stays outcome analysis. So this was really like a crown on the work that we did. And I think, in summary, a blueprint for successful product development is really that you need to define where you want to go. Where are you going to go, and what are the resources that are needed? How can you build a winning team? How can you get a team that is indeed able to excel and get with a little group so much work done in such a short period of time? So in my opinion, because you need a lot of money to get this done, as I showed you, you need a good plan, you need a team that is highly focused and that is passionate. And I think we were able to have that all. And as I said, I wanted to conclude with giving you some thoughts on the implications of our experience at Protein Sciences for a universal influenza vaccine, because people keep on wanting to develop a universal influenza vaccine, a vaccine that you can get once and will protect you to influenza forever. And I think it's going to be difficult, to quote Nancy Cox, who used to say, how can a vaccine do better than nature? I think she had a point there. The influenza virus changes so rapidly that it's hard to imagine how you can come up with something that will protect years into the future, and how are you going to demonstrate that? So I think from a product development standpoint and from a GMP manufacturing standpoint, it's easy. You can just define, this is what I'm going to do, and you can make that product, and you can develop a validated manufacturing process. That can be done. But I have some big red flags on clinical development, regulatory approval, and distribution. And just some food for thought, the clinical development challenge for a universal vaccine. There's no surrogate endpoint for performance. A clinical endpoint trial requires at least 10,000 participants, as the comparator needs to be a licensed vaccine in the United States, because today it may not be feasible to conduct a placebo-controlled study because this vaccine is recommended for the entire population, and therefore it wouldn't be ethical to do this. And how do you prove that your vaccine is going to be effective over multiple years? How do you deal with waning immunity? And are you gonna just basically show that in year 2, and you compare that again with a licensed vaccine where people now will have good immunity, how are you going to be able to show that you are at least as good as that? I think that that is a very important point that requires consideration. Another challenge, the regulatory challenge, is you really have to ask yourself, what is the desired indication? For whom are you developing this product? What is the duration of protection? The licensed vaccines that are on the market are considered safe and effective. This, you will require comparative efficacy studies. And, one can ask oneself, is there any value or acceptability of human challenge studies? And every clinical study that requires 10, 20,000 people will cost you at least $15 to $20 million. So where are you going to get the money from? So the distribution challenge was actually the challenge that led Protein Sciences to be acquired by Sanofi. Because even though we had a better vaccine, we were unable to sell it. And the value of the influenza vaccine market is very high. It's probably anywhere in the 2 to 4 billion. And the competition is therefore unlikely to give up this market, right? And I mentioned we were unable to distribute more than 350,000 doses in the United States, whereas Sanofi is now selling over 10 million doses. So with that, I want to open the floor for questions. Benjamin Franklin stated, energy and persistence conquers all things. So that is maybe, stopping on a positive note for those who are still trying to develop a universal influenza vaccine. With that, Indresh, I'll give it back to you.
Thank you, Mano, for very nice presentation. I think you covered a whole lot of ground in probably about 45 minutes. I think it's amazing. And again, I think it is from the development to the clinical trials to the challenges. We greatly appreciate. In terms of the questions, I think there are four questions. Number one question is, which cell line is used for the gene of interest expression?
So we use the cell line derived from the ovaries of the fall armyworm, which was Spodoptera frugiperda, and this was the cell line was adopted to be able to use under serum-free conditions and really optimize for growing into bioreactors. So Spodoptera frugiperda, or a derivative of SF9.
Yeah,
For those who are familiar working in that field.
thank you, Mina. The other question is, does the expressed HA generate hemagglutination in vitro? If so, does the hemagglutination level correlates with efficacy in vivo?
I do definitely. The expressed HA is a biologically active molecule, and it
Yep.
does generate hemagglutination in vitro. I think it is hard to say that it totally correlates, but what we saw in early clinical trials, and which is the reason why we ultimately went for a 45 microgram dose product, is that if you had more hemagglutinin, you would get a better immune response. And of course, you see that in vivo as well. But did we really do a very extensive, study to see whether that was a perfect correlation? Probably not, but I would expect there is.
Thank you, Mina. I think there's another question by Renato Astray. Are there immunological surrogates associated for suggested that explain the better efficacy of the recombinant vaccine over the traditional inactivated virus?
I don't necessarily think that there are immunological surrogates, because we have always wondered what was the main reason why Flublok was better, other than that it is a higher dose vaccine. But so is obviously Fluzone high dose, and yet the study that HHS published showed that Flublok still outperformed the egg-based vaccine. So are those egg-derived, egg-induced mutations, are they the ones that really cause Flublok to be better, because it's a really match with the circulating virus, right? So we don't have egg-induced mutations. Is it perhaps that the glycosylation of a hemagglutinin produced in insect cells is less abundant, and therefore, has certain sites that are important for your immune system better available? Is it the fact that you have perhaps more display of the stem and get some universal antibodies that is responsible for a better protection against drifted strains? I really don't know. I think we always speculated about it, but we cannot be sure.
Thank you, Manu. I think there is another question. This is a long one, so I will read it. And is there a resistance from regulatory perspective in approving a new vaccine platform when a vaccine already existed for flu? So I think if you want me to read the whole thing and then you answer, Manu, will that be right way?
Well, I think there's always resistance in approving a new platform. I think we have seen that with DNA vaccines and
Yeah,
RNA vaccines
RNA
prior
vaccine.
to a pandemic that really caused an enormous, response. There was no approved DNA vaccine or RNA vaccine, right? Even though many of those had been in development for a long time. So I think when something is new, there's always resistance because people will say we have this product that has a history of safe use. Why would we come up with a product that might not be as safe? And I would say that the FDA is used to be extraordinarily critical in ensuring that a product that you give to healthy people is safe and effective. And that's why, we were required to do these very extensive clinical studies.
So the other part of the question is there a challenge in clinical study design, considering that susceptible individuals will not receive the standard care, but but the test vaccine
I
probably
think
will.
that that is not a particular challenge because
Yeah.
everyone who's in a clinical study, they will receive weekly phone calls, they all get the same care, and obviously influenza is not necessarily a deadly disease, right? In principle, you can treat people as long as you know it early enough. So I don't think that that was a particular challenge that we faced.
So would the mRNA vaccine replace TIV or recombinant vaccines?
Well, I think not. But that's a personal opinion. I think
Yeah.
not because the side effects, like even though they might not be considered serious side effects, but the side effects after receiving an mRNA vaccine are definitely more aggressive than receiving a protein-based vaccine or an inactivated influenza vaccine. Just think about how many younger adults really had fever and all kind of actually had to stay out of work after receipt of the COVID vaccine. So therefore, I don't think that that is an easy hurdle to cross.
Well, Dr. Katani has paid a lot of attention to your talk. I think another question that is being asked is, would addition of neuraminidase ever fly as an added antigen to improve flu vaccine effectiveness?
Well, I definitely think so. I definitely think that protein sciences actually already had data that adding neuraminidase could help because neuraminidase doesn't seem to change as rapidly as hemagglutinin, but it also provides an enormous challenge. And if anything, if there would ever be, in my opinion, a advantage of a messenger RNA, it might actually be an mRNA vaccine for influenza. It might actually be adding that that neuraminidase in because the challenge that we faced when trying to develop a neuraminidase vaccine was that it was so hard to purify the product and to keep it active. And we didn't really know whether that activity of neuraminidase was necessary to maintain its immunological action. Because what a neuraminidase antigen would do is it would not prevent the flu, but it would reduce the spread of the virus should you still get infected, should you have a breakthrough infection. So it would, one, be a difficult clinical study, but I do think it would help if there is breakthrough.
All right, this is a question from our old friend Bob Hubner.
Ah,
So Bob is asking that, Menno, do you think that a more broadly effective flu vaccine needs to incorporate other flu antigens beyond HA? I think it's probably similar to
yeah,
using
I
NA.
think that's it gets covered, right, Bob, by the neuraminidase answer that I gave. I think it would be good. It would be of interest. But again, I yeah, I don't necessarily think that other antigens besides neuraminidase would be good candidates for that.
All right, Menno.
A couple
Other
raised hands, Indresh, if we
Yeah,
want to
yeah,
allow some.
absolutely go
Um,
for it. Yeah.
kind of so TFE 30.
TF830 is
Yep.
on mute, so
Yeah.
maybe we can just proceed with.
Okay. The next one is from Dr. Peter Halisi, so we can.
Hello, Peter. You're also on mute, but please.
Okay, Peter, can I start? Do you hear me?
Yeah, we hear you.
Yep.
Okay, a wonderful presentation. Manon, this was really great. I have just one question. You mentioned that the B component is slightly less effective as compared to the A component. And the same is true for the mRNA data in terms of the B virus hemagglutinin. Why do you think is that the B in your system in terms of the baculovirus, but also the mRNA, doesn't appear to be as good as the A?
That's a very good question, and I hope that Indresh is going to help me answer it. But I think it's because the B antigen is way more soluble. So in other words, I think our vaccine really worked very well because we formed these particles that I showed in the EM graph in the beginning.
Yeah.
And if you have a B antigen, it doesn't really do that very much. So there's something intrinsically with those hemagglutinin antigens from the B strain that doesn't work well. And maybe something like the clamp technology could help there. I don't know, Indresh, do you want to kind of chime in there?
Oh, well, Mano, I think you were right, but I think at the same time, I think we need to really compare them side by side and efficiency from the time. But that is one limitation that I see for the mRNA vaccine. For the purified vaccine, I think we did extra care in terms of characterization. So we are looking at the conformation, we are looking at the hemagglutination profiling, we are looking at the trimer formation. I think all these things are probably largely not done for the mRNA vaccine. So for the plant-based vaccine could be an alternative, but I think it needs to be looked at side by side.
Yeah. Peter, I hope that answers your question, but it's a complicated one.
Okay.
Yeah.
We know.
Thank you.
Absolutely. Ted, do you see any other hands, or I just keep on reading from the chat?
You can go back to the chat now. Thank you.
Okay. Okay, so I think the question is from Sujata Viz. Is the current Flublok only stable for 9 months? How does the stability compare with other vaccines?
I think the stability is probably very similar to other vaccines. And remember, the 9 months is good enough because, the next season has most likely another vaccine composition. So 9 months was all that we needed at that moment in time.
All right. So other question is from Messiel. I think being a vaccine with higher antigen concentration, Flublok is recommended to greater than or older than 65 years folks. But would you know if it is available for adults in US?
So that was the good thing about the Sanofi acquisition. They were able to get it everywhere. You can go to Walmart, Stop and Shop, CVS, but you do often have to ask for it because unfortunately pharmacists, if they don't really know the data behind a vaccine, they might actually give you the vaccine where they are getting the highest profit. And that might not be the case for the Flublok product that they have. But it's available for everyone above 18 years of age.
Thank you, Mano. I think there's a very interesting question by Safi Danieli. Is the protein trimer stabilized in some way or just truncated and secreted?
No, we definitely, we observed in early trials that if you make a secreted version of the hemagglutinin, it's not going to be immunogenic. It won't fold the right way. So you really need that transmembrane domain. And we therefore also purified the products from the insect cells themselves because they would be sticking through the membrane of the insect cells. So it's definitely a portion of the hemagglutinin, the transmembrane domain, that helps with the stabilization. And therefore, that is maybe one way to make the product better.
Yep. So there is another very interesting question by Dr. Abdul saying, great talk to you, Mano. Wouldn't a universal vaccine is developed has to be first approved as a seasonal one versus approved vaccines. Then clinical trial continue the next years, demonstrating protection, wanes seasonal vaccines, so it could be required long-term studies.
Yeah, I think that is a comment, right? And I would agree with that, because how can you claim that a vaccine is going to be efficacious over multiple seasons if you haven't demonstrated that?
So again, a question by Sudax Mordan. Did the hemagglutinin stay as rosettes in the vaccine preparation?
Yes, it did. Yeah. And I think
Uh,
the H3 had the greatest desire to form a rosette and to form a strong
yeah,
rosette.
yeah,
No adjuvant used,
yeah.
right? This
Yep, that's
vaccine
already
does not
there. Yeah,
contain adjuvant.
yeah. I think again, I think Sophie Danieli is asking, and also was the scaling production made with frozen virus or BIICs?
I
So
think
yeah,
so what we would do is we would create a frozen working virus bank, and that would be expanded. And of course, that is then no longer frozen, but indeed there was a process of freezing it down. And there's a book about baculovirus production by Springer that I edited last year, and that has a chapter in it that totally describes how to maintain those baculoviruses.
so thank you, Manu. Bob is asking, what do you know about the uh, decline in immunity of Flublok versus IIV over the flu seasons? Could this explain the better efficacy?
I think the decline in immunity is similar, right? It
Yeah.
just every 30 days, I guess, you see a decline, and it just is from that starting point in hemagglutination as you measure it, and then going down. Right. There was also a question I see about purification. I mentioned purified
Yeah.
to greater than 90%, but in principle, this product is 98, 99% pure.
9%. Yeah.
But we didn't claim that, because if you start claiming it, then you need to demonstrate that year after year. And the FDA was very helpful to us in really showing and describing, like, how do you do this so that from year to year you're going to be able to actually make a product, versus not being able to make a product because you were so you try to obtain a purity that was not possible for a particular hemagglutinin.
So there is another question by Nelson Marina. How much does vaccine effectiveness improve for people vaccinated previously against earlier variants? Which group of people do you recommend vaccinating?
I don't really understand that first part of the question, but I think, influenza vaccines, if you would give them to healthy adults, right, then you at least will prevent the spread of influenza. So I would always say I like to get my influenza vaccine because now I'm not spreading the virus, because the people that are most vulnerable for influenza obviously get that virus from someone. So I think the all-round immunization was a good thing that we had in the United States, and it is too bad that it's no longer officially recommended for pediatrics. But I don't understand the first part of that question. That may relate to the fact that I showed that if you were vaccinated during the previous season, you were not as well protected as when you were not vaccinated. So, I mean, you would probably want to do a study where you would study, like, what if we have an age group of 50 years and older, and we tell them we are now going to do a study to see whether you're actually protected for 2 years. But again, you need to, I don't know whether that's ethically feasible.
I think we are almost to the time, so we can stop. Ted, Menno.
Yeah, so I think what I would like to do with Indresh's permission and of course also Ted is basically share with you some slides that talk a bit about the ISV, the International Society of Vaccines. Indresh is the incoming president, and I was supposed to moderate his talk today, but we kind of as old colleagues turned it around. And thank you very much for moderating, Indresh.
Thank you, Mino, for the
And I'm looking
opportunity.
forward in 2 months. Indresh is again on the schedule. By that time, we should have been able to overcome the challenge that we face today.
Absolutely.
I would like to talk a bit about a number of activities that we're doing. It will only take 2 minutes for those of
Yeah,
you who have time to listen to that. So
yeah,
next week, we're
please.
going to have a mini symposium organized by Dr. Chime from Sanofi. He has put together an excellent panel of speakers, including Professor Osterhaus, Dr. Kisko from Sanofi, and Dr. Gravenstein, and they will speak about the human metapneumovirus. There has been a lot of developments around this topic. We have seen acquisitions first of Icosavax and then NextWaveBio recently, and Clover is also in all kind of talks. So this is really something that is on top of everybody's minds, I guess. So maybe next slide, Ted. We will also have a next generation vaccinologist webinar series. This happens every month, and in general, we try to take the first, I guess it's Wednesday of the month, or sorry, Thursday of the month. And this time it will, what we do in this series is we have an experienced scientist and somebody who's a little earlier in their career, and they talk about, in this case, it's going to be about TB. So really interesting conversation. Then we will have in February a seminar by Dr. Sean Liu, who was one of the founders of the International Society of Vaccine, and today is still, after many decades, working on an HIV vaccine, and he will share our progress because he has some really nice progress there. And I think that would be really interesting. So on our website, you can find a event calendar, and it really is good to look at that regularly because every month there will be a new, a new 1 or 2 new seminars announced, and every quarter we'll have a mini symposium. Okay, so
Yeah.
I would be remiss if I wouldn't thank not just everyone who attended this seminar today, but also our sponsors and our partners who enable the society to do what we do. We organize 1 annual congress. We also sponsor early stage career scientists and what have you to join our meetings. So I guess it's just a really exciting society to be part of, but thank you to all our sponsors. We have a lot of social media updates, and I think these QR codes can be used to scan. And of course, if you're not a member of the organization, we would invite you to join. We think that having, especially in this time, a society that really promotes science into vaccines is going to be useful, and so please join. Our Congress this year will take place in Belgium, in Antwerp, and it's going to be organized by Michael Schotsaerts, Lakshmi Krishnan, and Isabel Leroux-Rules. They're putting together an amazing program, and in our bi-monthly newsletter, we will provide updates on who the confirmed speakers are. And as I mentioned before, we have annual Congress awards for all kind of early career researchers and PhD students, and the applications will open in February. And that was it. So thank you all for your attention.
Thank you.
And thank you, Indresh, so much for modulating, and thank you, Ted and Lars, for helping and supporting. I know that there might be some more questions. Happy to answer them. If you email them to manoncox@isv.org, I guess. isvonline.org. Thank you, guys.
Thank you, Mano, and thank you the audience. Let's close here, and we'll continue to look for the exciting webinar series. Thank you.
Thank you.